Slide 1, an introduction to enzymes. Hello everyone, and welcome to our lecture on enzymes. My name is Muhammad Shahid Nadeem, and I'm excited to guide you through this fascinating topic. By the end of this lecture, you'll understand the basic definition and role of enzymes, be able to describe the 6 major classes, explain enzyme specificity and catalytic mechanisms, differentiate between simple and hollow enzymes, and recognize the industrial and medical applications of enzymes. Let's dive in. Enzymes are biological catalysts, essential for regulating metabolic reactions that sustain life. Unlike regular catalysts, enzymes are primarily proteins or RNA molecules. They accelerate reactions without being consumed, but they don't alter the equilibrium point, only the rate. In essence, life at the metabolic level is an orderly function of enzymes. The history of enzymes is intertwined with human civilization itself. Ancient Egyptians were using enzymes in cheesemaking as far back as 6,000 years ago without fully understanding their chemical nature. In 1878, Kuhne coined the term enzyme, and later, in 1897, Edward Buckner demonstrated that extracts from crushed yeast cells could convert glucose to ethanol, showing that living cells weren't necessary for the reaction. James Sumner, often called the father of modern enzymology, achieved a groundbreaking feat by isolating and purifying ureas in crystalline form from jack bean. Later, Sumner and Downs crystallized catalase from beef liver. In 1982, Thomas Sheck discovered the catalytic properties of RNA, suggesting that life might have even started with RNA. As our knowledge of enzymology grew, so did the need for a systematic way to name and classify enzymes. While older names like chymotrypsin and pepsin were retained, a common approach was to add the suffix "ase" to the substrate name, such as sucrase for sucrose. Another method involved adding "ase" to the reaction type, like hydrolyses for hydrolysis. However, these systems had discrepancies, leading to the need for a more comprehensive approach. To address the limitations of earlier systems, enzymes are often named based on both the substrate and the reaction they catalyze, such as malate dehydrogenase. In some cases, enzymes are named after the product they synthesize, like rhodonase. Another approach involves naming enzymes after the substrate hydrolyzed and the group involved, such as glycoside hydrolyses, which catalyze the hydrolysis of glycosidic bonds in complex sugars. Let's delve deeper into carbohydrate hydrolyzing enzymes. Beta-glucuronidases are part of the glycosidase family, responsible for breaking down complex carbohydrates. Human beta-glucuronidase, specifically, hydrolyzes beta-D-glucuronic acid residues from mucopolysaccharides like heparan sulfate. Now let's consider protein hydrolyzing enzymes, which act on peptide bonds. Endopeptidases, like pepsin and trypsin in animals, and papain and bromelain in plants, break internal peptide bonds. Exopeptidases, such as dipeptidases and tripeptidases, cleave amino acids from the ends of peptide chains. Renin, also known as chymosin, is a protein-digesting enzyme that curdles milk. Lipid hydrolyzing enzymes include lipases, esterases, and lysithinases. Lipases break down triglycerides into fatty acids and glycerol. Esterases hydrolyze esters into an acid and an alcohol. Lysithinases are phospholipases that act upon lecithin, a substance found in animal and plant tissues that attracts both water and fatty substances. In 1961, the International Union of Biochemistry, IUB, established a system to distinguish enzymes based on the chemical reactions they catalyze. This system divides enzymes into 6 major classes, each with subclasses. Each enzyme name has 2 parts, the substrate and the reaction type, ending in "ase." Additional information can be included in parentheses. Each enzyme is also assigned a systemic code number called the Enzyme Commission (EC) number. Let's break down the EC number with an example, EC2.7.1.1. The 2 inches represents the major class, transferases, 7 inches the subclass, transfer of phosphate, and 1 inch the sub-subclass, an alcohol group, as phosphate acceptor. The final 1 inch denotes the specific enzyme, in this case hexokinase, which catalyzes the transfer of phosphate from ATP to the hydroxyl group on carbon 6 of glucose. This slide provides a visual overview of the 6 major classes of enzymes according to the IUB system. We have oxidoreductases, transferases, hydrolyses, lyases, isomerases, and ligases. Each class plays a distinct role in catalyzing biochemical reactions. Let's start with oxidoreductases. These enzymes catalyze oxidation-reduction reactions, often requiring coenzymes like NAD or FAD. They facilitate electron transfer between molecules. Examples include alcohol dehydrogenase, cytochrome C oxidase, and catalase. Next, we have transferases. These enzymes catalyze the transfer of a group, other than hydrogen, between 2 substrates. Examples include phosphorylase, which transfers a glycosyl group, and hexokinase, which transfers a phosphate group. Hydrolyses catalyze the hydrolysis of substrates by adding water across the bond they split. Examples include lipases, which act on ester bonds, and proteases, which act on peptide bonds. Some proteases, like pepsine and trypsin, retain their traditional names due to long-standing usage. Lyases catalyze the removal of groups from substrates through mechanisms other than hydrolysis, leaving double bonds. Examples include aldolase, a carbon-carbon lyase, and fumarase, which is involved in the citric acid cycle. Isomerases catalyze the interconversion of optical, geometric, or positional isomers through intramolecular rearrangements. Examples include alanine racemase, which converts L-alanine to D-alanine, and glucose-phosphate isomerase, which converts glucose 6-phosphate to fructose 6-phosphate. Finally, ligases catalyze the joining of 2 compounds, utilizing the energy from the breaking of a pyrophosphate bond in ATP or a similar compound. Examples include acetyl-CoA synthetase, which forms a carbon-sulfur bond, and glutamine synthetase, which forms a carbon-nitrogen bond.